Welcome students. Previous class we have discussed about the structure of mRNA. Today we will discuss about the structure of tRNA. But before going to the structure of tRNA, here in the last class, that is in the structure of mRNA, few points are there left which I will tell you now. Here we have seen this mRNA is made up, that is it is an RNA which is formed from the DNA itself. Okay. So here all the exons were joined together, all the exons, that is coding parts, were joined together, they made the mRNA. All the exons, that is E1, E2, E3, E4, E5, all the coding parts were joined first, that is were joined together by an enzyme called as ligase. So this is the mRNA which has been synthesized from DNA itself. Okay. Now this mRNA is very unstable. To give stability to this mRNA, here that is at this is the 5' end, this is the 3' end. Okay. On this 5' end, a cap is added. A cap is added, and at this 3' end, a tail is added. A tail is added. With the addition of this cap and the tail, now this mRNA is stable. mRNA is stable. The tail is made up of polyadenine. The tail is polyadenine, that is made up of adenine molecules. And here this cap is methylated. It is made up of methylated guanine. Methylated guanine. So now with this together, all these together, this is the mRNA, and stability is given by this tail and the cap. So this is the actual structure of mRNA. This whole coding part, this is the structural gene. This is the cap and this is the tail. This coded part, which is nothing but, it is also called as, it is structural gene. So now this is the actual structure of mRNA, that is stable mRNA. So where this mRNA is synthesized? We said that mRNA is synthesized from DNA itself, but where it is synthesized? This mRNA is synthesized in nucleus. The DNA is in the nucleus, so mRNA, DNA is in the nucleus, and this mRNA is synthesized from the DNA itself. So where it is synthesized? mRNA is synthesized in the nucleus. mRNA is synthesized in the nucleus, and this mRNA works along with ribosomes. These ribosomes are present inside the cytoplasm of the cell. They are present in the cytoplasm of cell. Okay. So after formation, after synthesis in nucleus, the mRNA comes out from the nucleus into the cytoplasm. mRNA comes out from the nucleus into the cytoplasm. For example, this is a cell. In the center we have nucleus, and this is the cytoplasm. In the cytoplasm, ribosomes are present. Small units, that is ribosomes are present. So here this is the nucleus. Here in this strand DNA will be present. Now DNA will synthesize what? mRNA. DNA will synthesize mRNA. So mRNA is synthesized inside the nucleus. After its synthesis, it comes out from the nucleus into cytoplasm. It comes out from the nucleus into cytoplasm, and it works along with this ribosome. This mRNA from the nucleus comes out into the cytoplasm, and it works along with the ribosomes and tRNA in the synthesis of proteins. So this is the major function. So whatever the code was in the DNA, which was in the nucleus, so from the nucleus, that is from the DNA, it has taken the code, okay, and came out from the nucleus. Now it is helping this ribosome. With the help of this code, it is helping the ribosome and tRNA to synthesize the proteins. Okay, so first thing, it is synthesized in nucleus. Second, it works along with tRNA and ribosomes for protein synthesis. How it works, we will see when we see the structure of tRNA and how proteins are formed. Next is the next important point. The mRNA is short-lived. The mRNA is short-lived. It will live only for a shorter time. It will live only till it translated the information. So, whatever the information from the DNA it has got, till it translates it to the ribosomes or till it transfers it to the tRNA, till it transfers it to the tRNA, it will live till that time. As soon as it has translated that information on the ribosome, as soon as it has helped the tRNA, okay, so it will dissociate. It will dissociate. It is short-lived and it will dissociate after it has helped the tRNA and ribosome. mRNA is formed from the DNA inside the nucleus. After formation, it comes in the cytoplasm. It helps the tRNA and ribosomes. Okay, as soon as it has its help is over, then the mRNA will be dissociated. It will not live longer. It will live only till it has translated the information which is got from the DNA to the ribosomes. Okay, so it will only till that time live till that time. Afterwards, it will dissociate. So that is why mRNA is short-lived. mRNA is short-lived. Now, this was all about the mRNA. Now, this is the mRNA with the code for protein, code for synthesizing the protein. Now, coming to the structure of tRNA, structure of tRNA. Now, this tRNA is the smallest RNA. It is the smallest RNA, smallest RNA, and it is single stranded. It is single stranded, but it is not looking like single stranded here. This is the actual structure of tRNA. It looks like some double strand. It is not single stranded. It is single stranded. This tRNA is single stranded, but because of the pairing on this pairing of this nucleotides, it appears as it is double stranded. It appears clover leaf shape. Clover leaf shape. The shape of the tRNA is clover leaf. Okay, so it is as this. Here this is the 5 prime end, this is the 3 prime end. So, and this one is a little larger. This end is a little extended. This one, it does not end here. It is a little extended. Always at the end we have, that is CCA codes. That is these nucleotides CCA will be present, and at the end we have this OH group. OH group is present at the end, because 3' is there, it will be having OH group will be present, and this part, it is actually like this. So, because of this coding, it is appearing as double stranded, but it is actually single strand with 3 loops. It is single strand with 3 loops. This is the first loop, this is the second loop, and this is the third loop. It is a strand with 3 loops. The first loop, the second loop, and the third loop. Single stranded with 3 loops. It appears as double stranded because of the pairing of the same strand. This is the continuation only. This is single strand only. This is single strand only, like this. For example, if it is like this one, it is single only, it is not double stranded, but actually it is pairing with this ones now. So, that is why it is appearing as it is double stranded, but it is single stranded. Okay. Each tRNA has 3 loops. Each tRNA has 3 loops. So, the first is here, this is the 5 prime end, this is the 3 prime end. This 3 prime end is a little extended. It is a little extended. Okay, and here we have an OH group. As we have this OH group, it is also the place where amino acid, this is the site for synthesis of amino acid. This is the site for synthesis of protein, that is amino acid will be there here synthesizing. Here this one, this is called as the D loop. This is called as D loop. This is the stop codon, that is anticodon loop. I said you this tRNA has 3 loops. The first one, this is the first loop and this is called as D loop. This is the second one which is called as the anticodon loop, and this is the third loop which is called as T loop, which is called as T loop or GTPC loop. T loop or GTPC loop. Here GT, what is this P here? Okay, G is guanine, T is thymine, C is cytosine. What is P here? P here is pseudouracil. P here is pseudouracil. So that is why the name is that is GTPC. It is having guanine, thymine, cytosine and pseudouracil. So that is why it is called as T loop or GTPC loop. Here this loop helps in ribosome recognition. We said that ribosomes helps in the synthesis of proteins. So which ribosome? There are many ribosomes inside the cell. In one cell there are many ribosomes. So which ribosome will be helping in the synthesis of protein? So the function of this loop is to recognize that ribosome which helps in the synthesis. Like I said, many are there, red structures, many are there. Out of those many, which one is the one which is which has to help in the synthesis of protein? So this protein, which is which ribosome will be recognized by this loop. This loop recognizes the ribosome which helps in the synthesis of proteins, and this is the anticodon loop. Okay, we said that mRNA, this mRNA will have the code. mRNA will have the code. So now this loop will have the anticodon of this, whatever code is there, it will have the anticode on this one. So if this is the mRNA, so this mRNA will come here with the code. This is the mRNA with the code. So the mRNA from the nucleus, it will enter into the cytoplasm, then it will come near this tRNA. So when it comes near this tRNA, this anticodon loop will recognize this. What is this? The sequence here. The sequence here is AUG. This is the code. This is the code. So it will recognize its anticode. If G means, it will be C, because G always pairs with C. If U means, it always pairs with A, and A means it always pairs with U in RNA. So here it will recognize and it is the anticodon loop. It is the anticodon loop. So whatever code is there on mRNA, here we have the anticodon loop on this second one. And the third here, this one, this is called as D loop. This is called as D loop, which is also called as dihydroxy group. So this group helps in amino acid recognition, because for synthesizing proteins, these ribosomes require amino acid. So amino acids are many types. We have many types of amino acids. So which amino acids will be helping in the formation of that protein, that will be detected by this D loop. So this is the structure of tRNA in detail. In the next class I will explain you.